Good morning, everyone. So, as my title slide suggests, my SenSam research is primarily focused on microbial water quality, and we care about water quality for a number of issues. I've picked two pictures here to illustrate these. This is my little girl, Ella, and she's got her face in the fountain at the Botanic Gardens. So we care about water quality from the perspective of its safety for drinking and its safety for human contact. We also use water for a variety of applications in agriculture, in industry, and recreation. So there are many reasons we care about water quality, and shown here are a group of MIT students who are kayaking on Makritchie Reservoir, illustrating one of the reasons why we care about water quality in Singapore's reservoirs. So Singapore is a nation that is dominated by catchment area. 2/3 of the landmass is catchment, and this catchment is drained by approximately 7,000 kilometers of waterways that feed into 17 reservoirs that are situated throughout Singapore. And let's see, can you hear me if I'm over here? Okay. So these waterways and reservoirs are carefully managed by the Public Utilities Board, and the governing principle that they've put forward is the ABC program, and it stands for active, beautiful, and clean. The waters need to be safe for recreational use, active. They need to be beautiful, so aesthetically pleasing, both in terms of visual assessment as well as the smells and even the flavor. And importantly, they need to be clean. They need to be safe for human consumption and human use. So my research at SenSam has really been organized into three major thrusts. First is evaluating methods for tracking sewage contamination in the catchments. Second, we focused on looking at microbial activities in the reservoirs that can affect water quality. And third, more recently, we've started looking at the potential for top-down control of these populations by viral predators. So I'm going to walk you through some of the questions and the results that have motivated our research in these three directions. Now, just by way of overview, I'll let you know that my training is as an environmental microbiologist, and the methods that my group primarily uses are focused on extraction of environmental nucleic acids. So we collect samples from the environment, we purify DNA or RNA, and in some case we culture cells from the environment. And then we use these samples for subsequent analyses, such as using next-generation sequencing to generate to regenerate, I should say, the information content of the genetic material within the environment. And so from that sequence data, we can understand the diversity of microbes that are present in the sample. We can identify the microbes present in the sample, and in some cases we can design methods to quantify the abundance of the microbes in the sample using that information. And if we focus on analysis of the genome content or the expressed genes, we can also gain insights into the activities that the organisms are carrying out in the environment. All right, so I joined SenSam right around the 2-year mark of phase 1. And I teamed up with PIs Pete Shanahan and Lloyd Chua, who were PIs in SenSam phase 1, and they were looking at the issue of how to track sewage contamination in Singapore's catchments. Now, this is a little bit problematic because in the tropics, the traditional methods that we use in temperate environments don't work quite as well. I'm sure many of you have heard me talk before on this topic, but basically, in the temperate environments, we use E. coli as a marker for whether or not a water body is contaminated with sewage. In the tropics, E. coli can grow. So E. coli likes to grow at human body temperatures about 37 C, and we typically see temperatures in the range of around 30 C, which is perfectly permissible for E. coli's growth. So Pete and Lloyd and the students that they were mentoring at the time carried out work to look at the abundance of E. coli throughout the catchment and waterways, and they saw that the abundance was actually correlated to human activity. But they were recovering surprisingly high concentrations that were on the order of 1, 2, even 3 orders of magnitude higher than what you would expect in the US, higher than the GRAB sample limits in the US. So when I joined the team, I suggested we look at an alternative marker that targets an anaerobic group of bacteria that can't grow under oxygenated conditions, and that are thought to be symbionts of the human gastrointestinal tract, and therefore highly specific for human contamination. And so we selected the HF183 marker, which targets this Bacteroidetes organism, and at the time, this was kind of the best standard for considering these Bacteroidetes. And this marker is actually considered or used by the US EPA now for use in the United States. We saw that there was actually widespread detection of this marker throughout Singapore. So that was a surprising result, and it made us think whether or not this was actually a suitable marker for Singapore. I mentioned this marker had been developed for the temperate environments, and we asked the question, well, is this really a suitable marker for the tropics? So to address this question, we carried out a follow-on study where we took the samples where we had seen high levels of fecal indicator bacteria, including the HF183 marker, and we applied next-generation sequencing to determine whether there was a relationship between high levels of these markers and the proportion of sequences that were pathogen-like. So this runs you through kind of the details of the analysis. We sampled 18 sites, 2 different monsoon seasons, and we used next-generation sequencing to recover information from this 16S ribosomal RNA gene, which all bacteria have. It's a good marker for biodiversity. And we generated 1.2 million sequences. We applied algorithms for taxonomic classification of these sequences, which let us identify the bacteria at the level of approximately between genus and species. And then we categorized these into their similarity to known human pathogens, and we used the National Institutes of Health curated databases for that. We also classified whether our sequences were similar to bacteria that had been found in sewage samples in previous studies and in 2 samples that we characterized from Singapore. What we found from this analysis was that when we consider HF183, E. coli, land use, and collection month as potential predictor variables, a model of beta regression, which lets you model a distribution over an interval from 0 to 1, this model showed that E. coli was actually the best predictor for the proportion of sequences from pathogens or sewage-like taxa. And in fact, that HF183 wasn't a very good predictor for pathogen-like sequences, but it did predict the abundance of sewage-associated sequences. So this study just came out. This was part of the work of Jean-Pierre Nshimiyimana, who was a SMA3 fellow here at Sensam. And this was part of his thesis, and we've just published that. So our kind of initial motivating question, whether fecal indicator bacteria, in particular E. coli, was predictive for the distribution of pathogens, shows that in fact there is some information gained by looking at E. coli levels in the tropics. Even though the levels are very high, they still do seem to reflect the abundance or predict the abundance of pathogen-like sequences. So we can ask whether this is truly a proxy for sewage, or perhaps it's a proxy for similar environmental dynamics between the bacteria indicators and pathogen-like bacteria in the environment. So ongoing is a follow-on study using a second fecal indicator bacteria, Enterococcus. But I think this work underscores further need for reliable methods to identify human sewage contamination in the tropics. And this motivates our next study, which JP has done in collaboration with his advisor at the Singapore Center for Environmental Life Sciences, Stefan Wirtz. You're going to be hearing from Stefan next. And in this study, we asked whether tests for the human-specific Bacteroidetes marker could discriminate human from other fecal sources in Singapore. And in this project, JP collected about 300 samples, and these are samples from human volunteers, and then samples from a variety of animals, including some that are with particularly local flavor, like mynah birds and wild boars. So what he asked were, do fecal samples from humans, can they be distinguished from the fecal samples from animals? And he asked this question using several different tests targeting different Bacteroidetes markers, including the HF183 marker that we had studied previously, but also including some additional markers that had been developed and validated more recently. And again, I should emphasize that all of these markers are developed for temperate context, and really their application in tropical environments is understudied, and we just don't know how well they work in the tropics. Okay, so what we found was that the HF183 marker was actually one of the worst in terms of its sensitivity and its specificity. We did find, however, that two markers worked quite well in this data set. And so we've put this forward via a publication that's just coming out in Water Research as a suitable marker for use in Singapore. And so now this is available for ongoing water quality monitoring work. Now, in addition, we've also evaluated an emerging technology called digital droplet PCR, which is kind of an advanced method for quantitative PCR analysis. And we've shown marginal increases in the sensitivity and specificity of these methods. And this will be communicated via a forthcoming publication. Our feelings on this subject were that the improvement perhaps isn't enough to justify the current cost of the method. So this is an interesting conversation to have. And actually, I would suggest any of you who are interested stop by JP's poster, which is downstairs. Now, thirdly, we're interested in understanding how long these markers persist in the environment. So when you see them, does it mean that the sewage contamination is recent, or has it been there for a while, potentially? And this work is still in the process of being analyzed and formulated as a manuscript, but this is a picture that shows JP hard at work preparing his mesocosm samples, where he's looking specifically at whether these markers persist for long times in sediments. And one of the reassuring things we found was that, in fact, the Bacteroidetes markers don't persist that long. So if you see them, it indicates that there's been relatively recent contamination. All right, so the second major focus of my work is, again, on elucidating microbial activities in waters that can affect water quality. And so here you see depicted a picture of Kranji Reservoir. It's a really lovely picture. Note the lovely shade of green there. So this is algae, cyanobacteria. And it shouldn't be too surprising that Singapore's reservoirs have recurrent algal bloom problems because, like I said, these waterways, two-thirds of the catchment, or two-thirds of the landmass of Singapore is catchment. And it's a primarily urban environment, and so there's going to be things like fertilizer and traffic runoff that get integrated by rainfall and delivered to these reservoirs. And so it's a very understandable challenge to have, and you can see these blooms here. Here's a cyanobacteria concentrated in a plankton net. This is a surface scum that I took with my iPhone, like standing above it, about 5 feet above it. So some of the problems associated with cyanobacterial blooms are the production of algal metabolites that can be toxic. And this motivated my first interest in these blooms, which I pursued during SENSAM Phase 1. So we wanted to ask, what are the microbial activities occurring in the blooms, and in particular, can we see toxin gene expression? So on the heels of the work that I was doing with Pete Shanahan and Lloyd Chua, we also carried out a overnight sampling of the Kranji Reservoir. So we literally got off the plane from MIT, took a quick nap, and then headed into the field for this. We collected samples over a 24-hour cycle. We extracted messenger RNA, used next-generation sequencing to determine what genes were expressed in these samples. And what we saw was that, as we could visually tell by the scums on the water, the majority of those sequences came from cyanobacteria, in particular from Microcystis, which is a known toxin producer. When we looked for genes involved in toxin production, we saw that they were expressed in all the samples. And this was actually the first time anyone had looked into an environmental sample and verified that the genes were expressed in situ, so in the environment. Previous studies had looked at cultures in the lab, shown gene expression, but this was the first time we had actually seen the genes being expressed in the environment. And we noted that the sequences were higher at night than during the day, and we hypothesized that this could be due to a differential predation pressure. Okay, so now we have all this sequence data. What do we do with it? Well, this is an example of one of the analyses we used where we took a reference genome from a sequenced Microcystis aeruginosa strain, and then we compared the sequence that we got from the environment to that reference genome. Now, the reference genome is useful because the genes in it are all annotated with fairly high confidence. And so what we could do was determine what sorts of functions were being expressed in the reservoir. So this is just a list of the top 50 or so most highly expressed genes. And we can see that these functions, the highly expressed functions, correspond to things like buoyancy regulation, photosynthesis, DNA repair. And all of these things make sense within the context of a cyanobacterial bloom, where the cyanobacteria need to float on the water to effectively absorb solar radiation so that they can photosynthesize. So we asked the question, what are the activities of the microbial populations in a persistent cyanobacterial bloom? And we saw high expression of activities that made good sense to us: buoyancy, photosynthesis, DNA repair, and production of pili, which allow the scums to form. We also saw that between day and night, we could see differential expression of activities for photosynthesis and, surprisingly, for toxin production. And further, analysis of the other populations in the reservoir suggested an interplay between the coexisting populations. So cyanobacteria expressed a wide range of transporters for nitrogen uptake, indicating that they could use things like urea, which is commonly used as a fertilizer. And Proteobacterial phylum were enriched in pathways for biodegradation, and it's been suggested in other studies that proteobacteria might be key players in degradation of cyanotoxins and cyanobacterial metabolites, so this was interesting. And we also saw that the transporter profiles of the different coexisting taxa indicated some level of specialization. So some taxa seem to be more keen or attuned to metabolizing sugars, while others seem to be more protein specialists based on their transporter profiles. Okay, so with this baseline information of the activities in the bloom, we also realized that we had great there was another research group here in Singapore at NUS who were interested in similar questions, and so we shared our findings with them and we started to talk more about potential for collaboration. And this research group is a group of Professor Karina Jin at NUS, and this is her algal team here. And they were interested both in toxins, but also in metabolites that cause the water to taste bad. And this is a particular concern to PUB because people are more likely to call and complain when the water is not tasting very good. So this is some data that Karina's lab had produced and shared with us kindly last year. If you look into the waterways, you can see concentrations of these two compounds, 2-MIB and geosmin. The threshold of human detection is about 20 for 2-MIB, and some typical concentrations from a 3-day study saw MIB concentrations up to 300. So you could detect this by smelling, makes the water smell musty. So we wanted to get a better handle on the populations responsible for making these off flavors. And we collaborated on a series of monitoring programs, both monthly monitoring and diel monitoring, where the metabolite concentrations were compared to the cyanobacterial and microbial diversity. And this is an analysis that was recently published in Environmental Science and Technology, where we showed that the water body studied here was co-dominated by proteobacteria and cyanobacteria with some actinobacteria, which can also produce off flavors and odors. And we saw that through multivariate statistics, we could see that the cyanobacterial populations were more correlated to the distribution of these off-flavor compounds than were the other types of bacteria in the water. So this supported the notion that the cyanobacteria are responsible for producing these off-flavor compounds. We're exploring this further with metagenomics, and that allows us to put together a metabolic model for the bacteria in the sample. And we are also exploring this with isolate-based genomics. And our goal here is to figure out how exactly the microbes are making these off-flavor compounds. So this is an example of 2 strains that we just published genomes for. These both make 2-MIB. The one has a canonical pathway for producing 2-MIB by a known mechanism. The other lacks any genes for a canonical pathway, and so this raises the question, well, how is it making 2-MIB? Now, I'll just mention one more avenue of research that we're pursuing, and I'll invite you all to come down to our poster on this topic downstairs in the poster session. But we're also interested in how viruses regulate the abundance of microbial populations in the environment. And the main motivation here is that you can take a virus that infects a cell, and under the right conditions, you could potentially use that to kill off a bloom. And our idea here is that instead of introducing a new virus to the reservoir, can we just simulate the activity of the existing viruses? And so therefore, we're not dramatically altering the ecology, rather we're just helping these cyanobacteria basically to get a cold and drop down in population size. So we've done a couple of baseline characterizations for viral metagenomes in the reservoirs, and we have some work ongoing in the marine environment, and we're actually very excited to get the sequencing data pretty soon. And so please join us at the poster session. We can talk some more about viruses in the environment and other environmental microbiology topics. I'd like to thank my collaborators and the staff that I've worked with at CENSAM. CENSAM alums are Dr. Kevin Penn, Boon-Fei Tan, Martin Tay, and Jia Wang, who have gone on to establish careers in industry and working with the Public Utilities Board. Currently, I'm working with 2 recent graduates of the SMA3 program, Leiling and Jean-Pierre, as well as some great research engineers and postdoc Raji Kaushik, who has joined CENSAM relatively recently. And I'd like to acknowledge my collaborators, Professor Stefan Wirtz, Professor Karina Jin, Pete Shanahan, and Lloyd Chua. Thank you so much for your time. Thanks.
Oh, sorry, Masaki.
Janelle, thank you very much for sharing excellent data on the genomic indicators in water samples. My question is, could you briefly explain how are you going to link between the data on microbial source tracking markers and actual public health risks or infection risks in water bodies based on your study?
That's a great question. And actually, so public health epidemiology is a huge field. It's not my specialty. One of the main recommendations that came out of our recent ESNT paper was that we need to start adding next-generation sequencing-based approaches to these large-scale epidemiological studies where you look at risk of, you do prospective cohort studies of people who go swimming. They get you monitor how many gastrointestinal or respiratory illnesses they get, and then you relate that to the levels of certain markers that you've predefined, where we're suggesting that we start predefining the proportion of pathogen-like sequences as one of those markers. So, we're putting the idea forward in the literature, and I would be very interested to collaborate on such a study. The epidemiology part would need to be coordinated through a public health branch, though. Thank you.